7th मार्च 2009। आज नीट का लास्ट डेट है फॉर्म फिलअप का। तो मैंने तो फॉर्म फिलअप कर लिया था, ठीक है। देखते हैं लास्ट डेट, मेरा भी बहुत सारा बाकी है, अभी देखता हूँ क्या कर सकता हूँ। Okay, not only enemy scheduling replication would result into mutations. Furthermore, energetically replication is a very expensive process with deoxyribonucleic acids. Deoxyribonucleoside triphosphate serve dual purpose. In addition to acting as substrates, they provide energy for polymerization. Reaction. The two terminal phosphate in a deoxynucleoside triphosphate are high energy phosphates, same as in case of ATP. In addition to DNA-dependent DNA polymerase, many additional enzymes are required to complete the process of replication. With high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire. For DNA molecules, since the two strands of DNA cannot be separated in entire length due to very high energy requirement to the replication occur within a small opening. The replication occur within a small opening of DNA of the DNA helix, referred to as replication fork. Referred to as replication fork. The DNA dependent DNA polymerase catalyze polymerization only in one direction, that is 5 prime to 3 prime. This creates some additional complications at the replicating fork. Consequently, one strand, the template with polarity 3 prime to 5 prime, the replication is continuous, while on the other, the template with polarity 5 prime to 3 prime, it is discontinuous. The discontinuously synthesized fragments are later joined by enzyme DNA ligase. The DNA polymerase on their own cannot initiate the process of replication. The DNA polymerase on their own cannot initiate the process of replication. Also, the replication does not initiate randomly. Also the DNA replication does not initiate randomly at any place in DNA. There is a defined region in E. coli DNA where the replication originates. Such regions are termed as origin of replication. It is because of the requirement of the origin of replication that a piece of DNA, if needed to be propagated during the recombinant DNA procedure, requires a vector. The vectors provide the origin of replication. The vectors provide the origin of replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA. The replication of DNA takes place at S phase of the cell cycle. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA and cell division cycle should be highly coordinated. A failure in cell division after DNA replication results into polyploidy, a chromosomal anomaly. A failure in cell division after DNA replication, a failure in cell division, after DNA replication results into polyploidy, a chromosomal anomaly. You will learn the detailed nature of origin and the process occurring at this site in higher classes. Transcription. The process of copying genetic information. The process of copying genetic information from one strand to of the another DNA into RNA is termed as transcription. Here also the principle of complementarity governs the process of transcription. Except the adenosine complements now forms base pair with uracil however, unlike in the process of replication, which one set in the total DNA, which one set in the total DNA of an organism gets duplicated. In transcription, only a segment of DNA. Only a segment of DNA and only one of the strands is copied into RNA. This necessitates defining the boundaries. That would demarcate demarcate the region and the strands of DNA that would be transcribed. Why both the strands are not copied during transcription? Why both the strands are not copied during transcription has the simple answer. First, if both strands act as template, they would code for RNA molecule with defined sequence. Remember, complementarity doesn't mean critical. And in turn, if they code for proteins, the sequence of amino acid in the protein, the sequence of amino acid in the proteins would be different. Hence, one segment, hence one segment, hence one segment, hence one segment of the DNA would be coding for two different proteins. And this would complicate the genetic information transfer machinery. Second, the two RNA molecules. If produced simultaneously would be complementary to each other, would form a double stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become a futile one. Transcription unit in DNA is defined primarily by the three regions in the DNA: a promoter, the structural gene, a terminator. A transcription unit in DNA is defined primarily by the three regions in the DNA: promoter, structural gene, terminator. Promoter, structural gene, structural gene, terminator. Structural gene, and terminator. Transcription unit in DNA is defined primarily by the three regions. Transcription unit. Transcription হচ্ছে কি DNA to RNA defined by three regions in the DNA: promoter, the structural gene, a terminator. There is a convention in there is a convention. There is a convention in defining the two strands of the DNA, two strands of the DNA in the structural gene of a transcription unit. There is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity, the opposite polarity, the DNA dependent RNA polymerase, DNA dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5 prime to 3 prime. The strand that has the polarity 3 prime to 5 prime acts as template and is also referred to as template strand. The other strand which has the polarity 5 prime to 3 prime, the sequence same as RNA except thymine at the place of uracil, except thymine at the place of uracil is displayed during transcription. Strangely, this strand which does not code for anything is referred to as coding, which does not code for anything is referred coding strand. Ooh. ಈಗಲೇ ಕನ್ನಡ Neet. Bilmiyorum. It is DNA sequence that provides binding site for RNA polymerase. It is DNA sequence. It is DNA sequence that provides site for RNA polymerase. It is presence of a polymerase. It is the presence of oh, yes. Oh, shit, oh, when diabetes is mildly with regulation of change in expression. Okay, এটা হচ্ছে যে বাম। अमृतांजन मेरा सिर में दर्द और जब होता है तब देता हूँ। घूम को भगाने नींद को भगाने के लिए मैंने as if the neuron and the gene. It is a regulatory sequence that may be present in the other animal. Some other properties of this sequence shall be described when dealing with the regulation. The gene expression transcription unit and the gene is defined. Also, they gonna force it. Consider force it. Define a gene in terms of DNA sequence. The DNA sequence coding for tRNA or RNA molecule also define a gene. However, by defining a system